Cancer, a disease that has taken the lives of a countless number of people all over the world. We all know of chemotherapy as a type of treatment for cancer, but today I'm going to be talking about a form of gene therapy called CAR T-cell therapy, or chimeric antigen receptor T-cell therapy, where T-cells are a type of immune cell. One of the main reasons why cancer is so hard to treat is because of the fact that the cancer cells dodge the immune system. This means that the immune system does not see them as a threat and therefore does not try to fight them. But CAR T-cell therapy may be able to solve this problem. Now, let's take a look at the process. First, the T-cells have to be extracted from the patient's blood by a process called apheresis, where the components of the extracted blood are separated in an instrument that is designed as a centrifuge. The remaining blood is then retransfused into the patient's body. Next is the genetic engineering of the T-cells by using an inactive virus as a vector to deliver the genes into the T-cells. First, the capsid of the virus fuses with the cell surface membrane of the T-cells and then injects the genes into them. This will then be incorporated into its genome. The T-cells are now going to be able to transcribe these genes and synthesize the proteins they are coding for, which are specialized CAR receptors on the surface of the T-cells. Now, the T-cells can be put back into the patient's bloodstream. Here, they continue to multiply. Now, in the patient's bloodstream, specific CAR receptors are attracted to specific antigens on specific types of cancer cells because they're complementary to each other. Once the CAR T-cells are attached to the cancer cells, they're activated and can start to produce and release toxins that can kill the cancer cells. One of these is perforin. Perforin is a glycoprotein that is able to form cylindrical structures in the phospholipid bilayer of the cancer cells, which are hydrophilic on the inside and lipophilic on the outside. The pores formed as a result allow for the rapid entry of water and salts into the cancer cells. But for effective killing of cells, granzymes are also required. Granzymes are serine proteases. Serine proteases are enzymes that have the amino acid serine at the active site and are able to break peptide bonds in proteins. They're rapidly able to enter cancer cells via the pores produced by perforin. They seem to induce apoptosis, or programmed cell death, in cancer cells. Let's take a look at granzyme B. First, granzyme B cleaves or splits the caspase enzyme CPP32. A caspase enzyme is one that is involved in apoptosis. This activates the nuclease, or enzyme that breaks phosphodiester bonds in nucleic acids, called caspase-activated deoxyribonuclease, or CAD, by the controlled cleavage of the inhibitor of CAD. This inactivates the inhibitory effect of the inhibitor of CAD, which frequently invokes p53 signaling, and this ultimately kills the cancer cells. The CAR T cells then remain in the blood for a period of time to prevent cancer cells from returning.